Okay, welcome to the second speaker. So this short talk will be given by a tag team between Berkley and Aaron. Let me introduce Berkley worker. Berkley is an assistant professor at Michigan State University at DOE MSU Plant Research Laboratory. He earned his PhD at Washington State University and the postdoc with the USDA at UIUC before receiving 2 years von Humboldt Fellowship to the University of Düsseldorf. He is broadly trained in the biochemistry, genetics, physiology of photosynthesis, and has a special interest in fluxes of carbon and energy through photorespiration. Aaron Leipman, he is a professor of Department of Biology at Eastern Michigan University. He earned his PhD at University of Michigan and a postdoc at Michigan State University, where he received American Heart Association Fellowship. His research interests include identification of enzymes involved in the cell wall biosynthesis and to understand the physiological functions of cell wall polysaccharide. Please welcome Berkley and Aaron. Floor is yours.
Great. Thank you so much, Yasuo. It's great to be here with you today. Our two big goals for this presentation today are, one, to get you excited about the idea of photorespiration as an avenue for biocrop improvement, and also to give you a little taste of what we're planning to do with a recent award through the DNA synthesis program that's been complemented now by NSF funds. And we're early on in this, we're just starting, but we can paint you a picture, and hopefully in future years we can give you a more defined program as it progresses. So the first thing that I want to illustrate is that there is a tremendous amount of energy that's incident by the sun on our planet. This is a huge amount of energy. So if we added up all the energy hitting the area of Berkeley, California, over a 1-hour period, it would be enough energy to power a semi-truck around the world over 300 times. So it's a tremendous amount of energy. And when that energy is incident on a leaf, all of that energy doesn't actually end up as biomass. In fact, due to several biochemical inefficiencies in photosynthesis, we only end up with a narrow slice, around 1 to 2% of that energy that actually ends up as biomass. So we want to understand these inefficiencies so we can understand how they'll respond to future climates, but also imagine solutions, imagine ways to minimize these inefficiencies. Now, one particular inefficiency that we'll talk about today is photorespiration. And here graphically you can see it comprises a fairly large slice of that energy from the sun in the plants that haven't found a way to bypass photorespiration in some way. Now, to put this in a little more quantitative terms, if you added up all the energy or all the calories that went towards photorespiration in the American Midwest, looking just at the soybean and wheat crops, it adds up to about 150 trillion calories a year. Now, for pretty fundamental reasons, we're not going to get all these calories back. But if we could increase the efficiency of photorespiration or minimize the losses associated with photorespiration by just 5%, it would be worth about $500 million a year. Now, this is an illustration from a food system, but you could do similar simulations, and this impacts bioenergy crops in a similar way. And I should be clear here, this is bioenergy crops that haven't found a way to get around photorespiration. So these are crops like camelina or camelina or canola, for example. So what is photorespiration? Where does it come from? Well, we have this enzyme Rubisco, and this is the initial enzyme in capturing carbon to do photosynthesis with. When it grabs a carbon molecule in the form of CO2, we enter that carbon into the Calvin-Benson cycle or the C3 cycle, and we have carbon gain. On the other hand, Rubisco has a minor affinity for oxygen. When it grabs an oxygen, it forms an inhibitory compound that can't really readily be metabolized by the plant. And so photorespiration comes to the rescue. Photorespiration is the recycling process that recycles that inhibitory intermediate into something the plant can use. But like any recycling, it requires energy, and it proceeds with the loss of carbon in the form of CO2. So, and this is pretty fundamentally linked to the activities of Rubisco. Now, there's been a lot of efforts to try to improve photosynthetic efficiency using different modeling approaches. So with these approaches, they take all the reactions of photosynthesis. We've pretty well characterized how these reactions look and how they connect. Then they look up different kinetics of these different enzymes, looking at literature over the last 40 years, using kinetics from different species, oftentimes done at just single temperatures, sometimes measured under non-physiological pHs, and they put them into this network model, into a system of ordinary differential equations, and they probe that to try to get a better understanding of photosynthesis. Now, while that's been helpful and while that's been um, interesting to help guide improvement strategies, one thing that's missing is if you look at the parameterizations, they're not really robustly defined. When you have plants growing in conditions ranging from 10 degrees C to 35 degrees C, kinetics that were measured 30 years ago at 25 degrees C don't really capture that whole picture. And so our general strategy is to take a step back and to, instead of going with a very large, not as well parameterized model, we're dissecting out one component of photosynthesis, specifically photorespiration, and we want to develop really well parameterized models to run simulations of photosynthesis at many different temperatures and in many different species. Now, I'm not going to go into huge details, but here in red, so this is the basic reactions, here in red are different inefficiencies that may be introduced into photorespiration. And this gives you an idea of the type of approach we're using and what our goal is with going simple, going robust, rather than going large and not as well parameterized. So with that, I'm going to pass it to Aaron.
All right, hello everyone. So I want to just talk a little bit about photorespiration. So in on cell walls, which I picked up when I was at the PRL, way before that, when I was in graduate school, I actually did some photorespiration research, and so this project has been great because I'm getting to revisit some of my work that I did in graduate school. So photorespiration is a pathway that's involved in recycling the phosphoglycolate. So on the right of this slide, you can see this metabolic pathway, and you can appreciate that there are many enzymes that are involved in this pathway, and they reside in 3 different subcellular compartments, namely the chloroplast, and that includes 2 enzymes that we're studying, and then the peroxisomes with 4 different enzymes, 4 different activities, and the mitochondria with 2. And so these metabolites, so the pathway sort of begins in the chloroplast with the oxygenation reaction by Rubisco and producing phosphoglycolate, and through various metabolic enzymes, this is converted to glycerate, which then enters the C3 cycle. So there have been many labs that have studied this pathway and have identified the genes, the enzymes and the corresponding genes for them in the years since I was in graduate school. So we have a lot of information about the identities of these genes and proteins. And the ones that we're focused on are shown in the table on the left. So really, we're focused on these 8 reactions that I've highlighted in the model in the pathway on the right. So I'm going to tell you what we plan to do with those. So we have sort of a 2-part plan. So the first part of the plan is to express the photorespiratory enzymes individually and to tag these so that we can purify them using a mobilized metal affinity chromatography, and then to do kinetics analysis of the enzymes. Unfortunately, as I said, all of the genes have been identified, and many of these proteins express quite nicely in E. coli, and they produce active enzymes, or have in the past. And so we're just building on this work. We're planning to do kinetics that include testing the activity of these enzymes at lots of different temperatures, so that we can improve the modeling by having a much better idea of how these enzymes behave at temperatures. So the second part of our plan will then be to use the information that we obtain in part 1 to inform the models, and eventually we're hoping to determine or define maybe some new enzyme combinations that we can introduce that will improve the efficiency of photorespiration. And this, as Berkley indicated, could have profound impacts on the productivity of plants. All right. So so the JGI has constructs, or is in the process, has made some constructs and is producing some more to feed into these plans. So we have asked or requested the photorespiratory enzymes be produced in expression constructs from a bunch of different plants, including Arabidopsis, which is we're using sort of as a model proof of principle, and then a bunch of different crops, including Brassica rapa, Camelina, this red alga that grows at high temperature called Cyanidioschizon, soybean, tobacco, and another warm adapted plant called Rasia stricta. So we're hoping by studying photorespiratory enzymes from plants and algae that grow in all sorts of different conditions that we may find different enzymes with different profiles that may be particularly useful or beneficial for photorespiration. Next slide, please. Oh yeah, thank you. So where are we in the project? So we have obtained already the expression constructs for Arabidopsis photorespiratory genes from JGI. And we have requested all the rest, and they are in the process of being synthesized right now. And we have expressed a number of recombinant Arabidopsis photorespiratory enzymes in E. coli already, and purified some using IMAC. So next slide. You can see here's an SDS page, image of an SDS page gel showing that we have successfully expressed a whole bunch of different ones in E. coli, and we're getting a lot of protein, and we're using it to purify. I'll show you on the next slide a purification. So here's IMAC purification of just one of these, Arabidopsis GGAT2. This is a glutamate glycosylate aminotransferase that's found in the peroxisomes, and you can see that we can get a bucket load of recombinant protein that looks pretty pure just after the IMAC. So I think we're on our way. We've got a lot of proteins that we're going to be working through, but we're kind of establishing the conditions that we're going to hopefully just be able to apply for the whole group of them, and we'll be having a lot of proteins and a lot of kinetics to do soon. So yes, so we're currently the scaling up stage of this and getting ready to do a lot of purification and characterization, and hopefully by the next JGI meeting we can fill you in with some of the some information that we found, and maybe some of the modeling that we're able to do with that information.
Great. So that's us. That's our project so far. Want to give a big shout out to Han Bao, who's a postdoc who worked a little bit on the project initially, and then now Ludmila Rose, postdoc on the project currently in my lab. And do you want to introduce anybody on your side?
So Meyer is an undergraduate who's been working with me on purifying the aminotransferases. So yep, yeah, that's my team right now.
And then recently, since starting on the project, we found that there was a pretty advanced effort going on to model photorespiration going on at the University of Dusseldorf, where I had done a postdoc. And so rather than try to come up with competing models, we're basically building off the same bones. We do have a collaboration now with Heinrich Heine University in Dusseldorf to try to come up with a really well parameterized and robust model. And so we're happy to be collaborating with them. And then we're happy for the support of the JGI Institute, as well as an NSF Foundation Award to do this project specifically, and then general support that I get through my affiliation with the DOE PRL here at Michigan State University. So thank you so much.
Thank you very much, Berkley, Arlan, for the great talk. We have one question on chat from Matthew. To what extent are the enzymes involved in the large complexes in vivo with metabolite shuffling from enzyme to enzyme? How can these be modeled if you are examining the recombinant enzyme individually?
Yeah, that's an excellent question, Matthew. And maybe you're familiar with this idea that metabolite channeling, specifically in photorespiration, seems to be a very prominent feature, and seems there's some good evidence that it does happen to some degree. So initially, we won't start down that road, and we'll just see how well the model does at representing measured metabolite data without factoring that in. If we do need to bring that in, there's going to be different kind of tweaks that we can do to how we handle substrate concentrations and how big we make the compartmental size that these enzymes exist in. And so I think we have some approaches to address that, but we'll just kind of see how well a simpler model works and then add complexity as the data indicate we need to add that.
Okay, so time's already up. So let's thanks Berkley and Aaron again. Thank you very much for the very nice talk.
Thank you.
Thank you.
Thank you.